online, a great crowd in person here at the Shady Grove campus at University of Maryland. Welcome all to this great workshop. This is the very first workshop for 2025 for CRCG. We're great to see all of you. I'm Visha Krishnan. For those of you who have not met me, I'm the Associate Director for the Center for Research on Complex Generics. In a moment here, Jim, our co-director, is going to be here to give the introductory remarks. He'll give an overview of CRCG for those who are not familiar with the center. All that I would like to ask you is if you've not connected with the center, reach out to us. We'd love to have a conversation with you if your company is not spoken to us. We're always interested in learning what your companies are up to, what kind of pipelines you have, what kind of challenges you face in the development of complex generics. Just a few housekeeping things for people in the room. If you're looking for restrooms, look for the signs at the top. When you leave the room, it's on the left. Coffee is at the back. Your name tags would have labels. The colors mean something, and that is for the in-person only discussions. So we'd be breaking out into groups later, and you'd be meeting people who have the same colors on their name tags for your group activities. I know several people had that question before. And for those who had ordered lunch, the lunch will be delivered to this room. I know some of your receipts and invoices said you'd have to pick it up or something, but you don't have to go anywhere. Lunch will be here. It'll be right back at the table. With that, I'd like to invite Dr. James Polli to give the opening remarks. Thank you.
Okay, good morning, everyone. It's really wonderful to be together in person. It's really fantastic. So my name is James Polli, faculty member at the university. Tim, we need a video up front. Video, yeah. My name is James Polli. I'm a faculty member at the University of Maryland. On behalf of myself and Anna Schwendeman, also co-director of CRCG, welcome. Welcome to this workshop on IVPT. Implementing FDA's IVPT guidance recommendations, a step-by-step illustration. Okay, so here's a listing of our goals as CRCG. I think some of you maybe have been at some of these meetings in the past. So it was established in 2020. The CRCG is a collaboration between the universities of Maryland and Michigan and the FDA. Really, our goal is to increase access to safe and effective generic medications through enhanced infrastructure and communication, education, research collaboration across industry, academics, and the FDA. We're dedicated to advancing programs that stimulate scientific dialogue. That's why we're here. It's important to be here to discuss things. And IVPT is probably a pretty good example of why we sometimes need to get together, since there's nuances in the technique, to disseminate current insights and generate new knowledge about complex generics in support of FDA's mission to promote and protect the public health. The American public benefits from the availability of generic drugs. The work you do, the work we do, the work that the FDA does is really all vital to ensuring continued access to these essential therapies. Generic drugs are essential to the affordability, sustainability, and reliance of our healthcare system. Generic drugs represent more than 90% of all prescriptions filled in the US, and yet account for only a fraction of total drug spending. Of course, complex drug products can make it difficult to identify bioequivalence methods and standards other than clinical endpoint studies. But of course, clinical endpoint studies are knowingly undesirable testing options in terms of sensitivity, testing efficiency, and testing cost. Overall, some might even say that the requirement of clinical endpoint studies can even be a disservice to patients in terms of medication access. So efforts to better understand drug product performance and identify scientifically sound bioequivalence testing that reduces regulatory burden is welcome really by all, right? Patients, caregivers, and manufacturers, as all manufacturers rely on the bioequivalence standard to maintain medication availability. Everyone here today appreciates the opportunity to discuss and make IVPT techniques an alternative to clinical endpoint testing a more reliable technique where best practices are more uniform. So here's, as I kind of said, some of our goals. Really a big part of what we do is we try to do select research and working with the FDA. And we also spend quite a bit of time talking with the industry to better understand problems they face and have workshops similar to this to really move the science forward and bring generics forward. Again, we really appreciate being able to interact with all of you and hopefully bring you some programming that's very helpful, such as today's programming of this two-day meeting. And here is just a representation of some of the companies that we've done outreach over the last couple of years. Appreciate being able to speak with you about generic drug development. So just in terms of IPT, we've actually, our first meeting that we put on several years ago was about IV RT and IVPT, and if you go to our website, complexgenerics.org, you can still have access to that multi-day event. All the notes and presentations are still available. FDA, SBIA also had a workshop soon after the guidance that was issued in 2020, and the guidance is referenced there. So in general, just in terms of where we've been the last couple of years, we've had 17 workshops, CRCG workshops and training with, I think we'll probably exceed 35,000 registrants by the end of today. And just some notes there in terms of this coming fall, we have a couple workshops in the fall, one on particle sizing, one on ophthalmics, and one on, I have a hard time reading the monitors out here, and one on AI in generic drug development. We have several research projects on the go. I think one that this audience might be particularly interested in is, a toolkit the last entry there, toolkit to assess adhesion performance of topical products. I'll also just say that we did contribute a little bit to, the FDA recently did issue a update of their guidance on guidance that involved, potential use of antioxidants for mitigating nitrosamines, and we're happy to contribute some of the science to that. Again, here's part of the team. I think maybe some of you have interacted with some of us before. So Visha, we just heard from this morning, our manager. Dana's out front, and Jen is online. I'll just ask Jen, just say hello to Jen, and also to remind everyone online, since we do have a large cohort online, that these notes from this meeting and the videos from this meeting will be available within 3 weeks. And everyone who's registered, we will email you and let you know when those, when that's all been posted to the web. Okay, and I guess one suggestion I would have for you interested in CRC, just kind of, we do periodically put out some emails, and the best way for you to receive emails is just sign up for our listserv, at least in terms of some of the updating programming, which our next one is on particle sizing in September. Okay, so big thanks to the folks on the planning committee, many of whom you will hear from today. Big thanks to Priyanka, who I'll introduce shortly. Also, really just a huge thanks to the vendors next door. The vendors, we've been very lucky to have several vendors be able to come here and set things up so you'll have an opportunity to interact with the vendors in terms of what equipment is available to do IVPT from Logan, Permagear, Teledyne, Cyberderm, and Delphin. Really coming all the way from Finland. Really appreciate all their efforts in supporting this dialogue. Okay, so thank you for your participation. Really glad to be with you. And what I'd like to do is introduce two people from the FDA. I'll first introduce Priyanka, who will be up in a few moments. She's, in addition to help really putting the whole program together, she's also leading this first morning session. So Priyanka is a lead. I have to take off my glasses. And I'll just read some, there's more detailed biographies on the web. Priyanka is a lead pharmacologist within the Division of Therapeutic Performance, ORS, OGD. Her areas include products in the topical and transdermal drug delivery area. She is responsible for facilitating pre-application requirements of generic drugs by conducting and promoting regulatory science research and to establish standards to ensure therapeutic equivalence of new drug products. So I think in the topic layer, I think you all probably know Priyanka. I'd also like to introduce Markham Luke, who will be giving our morning presentation. So Markham Luke serves as FDA Supervisory Physician and Director of the Division of Therapeutic Performance 1 within ORS OGD. His group is responsible for facilitating pre-market development of generic drugs through the regulatory science research. He has an MD and PhD in pharmacology from Hopkins and completed his dermatology residency and fellowship at WashU in St. Louis and NIH. He's been at the FDA since 1998, including lead medical officer for dermatologic drugs, chief medical officer and deputy director of the Office of Device Evaluation in CDRH, and as acting director of cosmetics in the Center for Food Safety and Nutrition. Thanks for being here, Markham. Well, we look forward to your comments. Thank you for being here. That's not quite working, unfortunately.
That's fine. Yeah. We'll look at the screen over there. Thank you, Jim. Thanks for the kind introduction. And good morning to all of you. Some of you had traveled a bit to get here. Really nice seeing everyone. We have some stars in the field of IVPT, and including folks from FDA who've worked hard to put this together with our collaborators in CRCG. I really appreciate all the work that went into putting this meeting together. I'm going to talk a little bit to introduce the topic. IVPT stands for in vitro permeation testing, and you'll say permeation of what? And we'll talk a little bit about what it is. First of all, like Jim mentioned, I'm a dermatologist. I'm board certified in dermatology, and this is highly relevant to the care of patients with skin disease. And we'll talk a little bit about some of the clinical relevances of IVPT. So what is IVPT about? It's about topical drug products applied to the skin. And here is a picture of a cream. And so IVPT is generally very useful for products with mixed phases, things that are a little more complex in design for topical products. This is a picture of the skin, not super close up, but enough that you can appreciate that skin is not exactly flat. It has sulci and gyri, and there are tiny hairs in it, and your skin is varied. It's the largest organ in your body. And it provides a good barrier to keeping things outside of your body. Like it protects your body from a variety of things, including foreign organisms, substances, toxins from the environment, et cetera. And without it, you would be in some amount of trouble. And keeping it intact is key to ensuring the homeostasis of the human body. But with that goes to, if you're trying to provide a therapeutic drug to the site of action, the skin also provides a barrier to permeation of those drugs. We'll talk about IVPT. IVPT is not designed to measure the penetration of the drug to the skin, but rather it serves as an approach to doing, for us, comparisons of one product to another, and seeing what the differences in rate of penetration could be. There's a little further breakdown in a more microscopic and 3D Frank Netter type drawing of the skin. And you see the skin layers, the epidermis is up top. That's the closest to the surface, down and with the dermis below and the subcutis below that. Largely the barriers are present mostly in the epidermis. The epidermis has a stratum corneum in it, which and tight junctions. There's a lot of things intended to keep water, keep chemicals outside of your body from penetrating in. There are blood vessels that crisscross in through the dermis, and those are designed to carry products to and from the skin as an organ and keep it and nutrified, I guess is the word. And you can also see hair follicles. The hair follicles actually penetrate through the epidermis around and somewhat into the dermis. So you can see and the subcutis. So you can actually see hair follicles going deep through the skin. So that presents a variety of additional challenges to looking and modeling actual skin penetration. And as you and I know, different parts of the skin in your body have different amounts of follicular amounts, and some of it is a little more follicular compromised than others, and the scalp, etc. But it's part of being who we are, and we appreciate the diversity. So IVPT, if you look it up on a variety of dictionaries, is a laboratory test used to measure the permeation of active pharmaceutical ingredients through the skin, attempting to simulate real-world conditions. And this has only been in the dictionary for maybe the last 2 decades. It's a relatively new technique. It was invented for a specific purpose, and some of that will be discussed through the course of these 2 days. It may be used comparatively with different products to demonstrate bioequivalence between some generic and reference topical drug products. And that's the intent of how we use it at FDA in the Office of Generic Drugs. And this is the focus largely of this conference. This is one of the devices that started this. This is the Franz cell, thanks to Tom Franz. And you can see there's a part of it is where you put the membrane or the skin, and you then can apply drug up on top. And then down in the bottom, there's where the receptive fluid goes. And I'm not an expert in the use of the Franz cell, but there are folks here that are. And they'll be talking more about this and also about how it's integrated into more modern products to look that are designed actually to do comparisons and do multiple samples, etc. Again, this is a skin, so skin can be separated. How you separate the skin matters, and folks have tried using different separations of the skin, acquiring skin for their laboratory testing. How you store the skin matters. If you freeze the skin, you may destroy the barrier to some extent, but in a comparative test, it may not matter so much. So some discussions that are going to happen at this meeting is what are appropriate preparations of skin as you prepare for a use in vitro permeation testing. I want to bring back to the drugs. So Q1, Q2, Q3 are well known to most of us. Q1, these are descriptions in shorthand for product for the topical product that we're looking at, or it could be applied to any pharmaceutical. Q1 is the question of what is the quantitative composition of the product, and if you're comparing 2 products, are the Q1 the same? Are they quantitatively the same? Or are they qualitatively the same? I think I got those mixed up around. It actually should be Q1 is qualitatively the same. So, and then Q2 should be quantitatively the same. And then Q3 is the physical chemical structure. And then sameness is defined by the measurement standards. There's some measurement error allowable, including batch variability in the product. So just want to put that together. And what results in how do you get batch variability? And that largely goes to the manufacturing apparatus for the products. And this is a mixer. So topical products are thrown together by putting a lot of excipients together, including emollients, whether it be there's water, there's petrolatum, etc. And they're all mixed together at various rates, various temperatures, and for varying amounts of time. And with that adds to some variability in the product mixture. And that adds to the notion of what how do you get to Q3 from Q1, Q2? Q3 is the basis also for the look and feel of a product. And with emulsions and mixed phase formulations, there's greater degrees of freedom for those products. These include considerations for particulate and globules, viscosity, and also metamorphosis, the absorption and drying of the product, as well as where does the drug compartmentalize within the emulsion, whether the oil phase or the water phase, for example. This is a photo of someone spreading the product on the skin. How you spread the product matters, the amount of product. Sometimes we see protocols where they say, okay, we apply 1 milliliter per square centimeter of skin. That's quite a bit of product. So, and is that relevant for application of a product to skin in real life? And I would say that we want to look at some of those a little more carefully, but also in a laboratory test, carefully thinking about how a product is applied to the sample skin is important to think about. So why is IVPT important clinically? IVPT evaluates comparatively the permeation of the active ingredient across a sample of human skin, a bioequivalence assay. It's important for non-Q1 same, non-Q2 same formulations, and provides bioequivalent parameter that considers skin permeation, especially important for these multi-phase topicals that have site of action in the skin. It ensures that generic products apply the same therapeutic effect as branded products, especially in terms of absorption and drug delivery. I just want to note that if the site of action is beyond the skin, or some of the site of action beyond the skin, then the blood concentrations of the product as the product penetrates the skin, that may play a role as well for bioequivalence. And so we may factor into that as well in that determination of bioequivalence. Again, close-up of someone spreading the skin. People have different techniques of spreading the products on the skin. And also it matters if, for example, the hydration of the skin to begin with when you apply to skin. Like when I prescribe products to patients, sometimes you'll tell them, apply this after you shower so your skin is very well hydrated, and you then have like a petrolatum barrier, especially for ointments, to apply over that skin, and you maintain that some moisture in the stratum corneum. But if you apply it to dry skin, it may actually absorb whatever moisture is in the product itself into the skin. And so the actual state of the skin can matter. So you can see people take care of the skin differently as well, and they also use other products in the skin. So in real life, there are some variabilities to how products are used. So bringing back to the workshop, our workshop goals, we'll have a description of IVPT method procedures, some of the challenges and some of the solutions. We'll be discussing what are common challenges. We're also going to get a treat of looking at step-by-step processes of IVPT studies, including some practical demonstrations, thanks to some of our suppliers and also to some of the experts in the use of IVPT. We'll also be discussing critical considerations and best practices. These include considerations for method development and validation, best practices for handling issues with IVPT studies. As I mentioned, the skin sample variability, challenges in handling formulations, and apparatus inconsistencies. Of note, skin sampling, much of skin is often available from skin flaps used from plastic surgery. That's where you get the biggest chunks of skin for products, or from cadavers donors. And then there are other sources of skin, some of which may not be entirely suitable for IVPT. And I think that's it. That was it for my slides. And thank you so much. Look forward to meeting all of you in person and having some good discussions about this important topic. Thank you. Next, we have Priyanka. Priyanka is is a star team lead in my division. Priyanka's worked hard over the years to really establish the topical drug product landscape in the generic drug field, and we're actually really proud to have her working at FDA with us. And Priyanka, come on up.
Hey, hello everyone. Thank you so much for joining us both in the room as well as online. We are very, very excited to have you. So I'm just going to take a couple of minutes to talk about why are we here and what are we going to talk during day one of this workshop. Over the last few years, since the publication of the cyclovir guidance and subsequently the IVPT guidance, we have received a lot of pre-NDAs as well as ANDAs that contain IVPT studies. And in those submissions, we have observed some challenges which appear to be related to either method development, method validation, related to variability in the data, aberrant data. So we are going to talk about all of these within the scope of the next 2 days. The challenges that we have observed suggest to us either there are some challenges with interpretation of the guidance, like what are we expecting when we say something in the guidance, or related to implementation of the guidance with your specific instrumentation, especially some of the more automated type diffusion cells. So since IVPT studies are an important component of our overall characterization-based BE approaches, which I'm going to talk about in a little bit, this does translate into lack of availability of generics for certain products. So our key goals for the workshop today is we would like to understand from all of you what are your key concerns related to the design and conduct of IVPT studies, as well as related to interpreting the data when it comes to the IVPT guidance. And an ideal outcome of this workshop for us would be if by the end of this workshop, we have a shared understanding of how to develop an IVPT method. How do we know that a method has been adequately developed before we move that method into method validation and pivotal studies, and of course, data analysis. So with that in mind, for day one of the workshop, for session one, we are going to talk through the considerations and the data that supported the development of the IVPT guidance, as well as some of the challenges that we are seeing both from an FDA and the industry perspective. In session two, we are going to systematically walk through considerations related to IVPT method development, both theoretically, how do we implement it in our labs, and practically using demonstration videos so that we have a shared understanding of, again, how do we develop an IVPT method. And last, for the in-person attendees in session three, we will have a hands-on activity and other equipment demonstration so that we can together develop an IVPT method for a hypothetical product and familiarize ourselves with some of the equipment. Before I introduce our moderator for session one, I did want to mention that we have a lot of attendees both in the room and online, and we encourage you to either type your questions in the chat or come to the microphones which are present across the room. And we will do our best to try to get to as many questions as we can. However, even if we don't get to your questions, please do send them because we plan on triaging all of your questions to really understand where your concerns are and how we can incorporate or clarify those in future versions of the guidance. With that, it is my pleasure to introduce our moderator for session one, Dr. Tannaz Ramezanli. Dr. Ramezanli is a senior pharmacologist within the Office of Research and Standards within OGD. She specializes in topical, transdermal, transmucosal products and leads development of general and product-specific guidances, reviews and response to control correspondences, citizens' petitions, and leads our strategy for industry meeting requests. She also leads the research within GADUPA regulatory science initiatives that supports the development of some of our guidances. Prior to joining the FDA, Dr. Ramezanli received her PhD in pharmaceutical sciences from Rutgers University and PharmD from Tehran University of Medical Sciences. So please join me in welcoming Tannaz.
Good morning, everyone. Hope you're enjoying the workshop so far. As Dr. Ghosh mentioned, in session one, we have 2 FDA speakers and 2 industry speakers. The first 2 talk from my colleagues at FDA We'll cover how the recommendations in the IVPT guidance were developed and what research supported those. And then you're going to hear about an overview of the ANDAs in which IVPT was used as part of the characterization-based B approach, and their status, and some of the challenges they had for approval. And then we're going to hear from two industry speakers where they talk about their experience with conducting IVPT studies and their challenges with implementing the IVPT guidance. So with that, I will introduce our first speaker, Dr. Sam Raney. He is the Associate Director for Science and Chief Scientific Advisor at FDA's Office of Research and Standards within OGD, where he orchestrates the GDUFA research program for generic drugs. He has over 30 years of experience in pharmaceutical drug development, specializing in topical and transdermal products. He has produced numerous research manuscript, review articles, book chapters, and patents. He has been a researcher and adjunct professor within academia, a principal or investigator on over 400 pharmaceutical product studies, and he served on multiple expert committees and panels for USB. Dr. Raney holds a bachelor's degree in molecular physics and biochemistry from Yale University and a PhD in biochemistry and molecular biology from University of British Columbia in Canada. Dr. Raney.
Thank you so much, Dr. Ramezanli. It is a pleasure to be with you here today, and I want to thank all of you here in person and so many of you online who have taken the time to come, and your interest in this field. It's very heartening. So this workshop is focusing on the implementation of FDA's guidance on IVPT, because it is a challenging methodology, and there's certainly been some challenges with implementing the guidance. So perhaps it's natural to begin with a talk about, well, where did these guidance recommendations come from? So I'll offer that talk for you today to give you a little bit of insight into where these recommendations came from and how they were developed and why it is that we have these recommendations. Let's begin with talking about what is an IVPT study, only because thank you also for answering the survey that the CRCG put out. It gave us a much better understanding of who you are in the audience today, and it is a very even distribution of experience and skill sets from novice to expert. And so I want to make sure that that this is meaningful for all of you. I'll begin at the beginning with what is an IVPT, and really what is an IVPT study design. So it begins with the human being, the donor, as we call them, whose skin is used in the study. So skin is taken from the body and used off the body ex vivo or in an in vitro system. We typically take multiple replicates of the skin sections from the same anatomical region, right adjacent to each other typically, because there is micro-regional variation in the skin, which both as a biological tissue and because our skin receives insults throughout our life in different ways in different regions, there is micro-regional variation, and this gives us a much more accurate representation of the bioavailability and the permeability characteristics of the skin. That skin then, each section is mounted onto a diffusion cell where the drug can then be applied. In this case, the test product is applied on the skin, mounted on the diffusion cell. And the skin itself, the primary permeability barrier is the stratum corneum. If you've ever had a very bad sunburn and peeled, that very thin layer that peels off is the primary permeability barrier for the skin. And similar to your nails or your hair, it is a non-living barrier, so that even after death, the barrier properties are maintained and can be studied in this in vitro system. So as the product is applied on the surface of the skin, It permeates through the skin and can be collected, accumulating in the receptor solution, which is typically a physiologically based solution. And then that can be sampled at different points in time to provide a pharmacokinetic, a cutaneous pharmacokinetic profile characterizing the rate and extent to which the drug becomes available at the site or sites of action in the skin. And often the skin that's mounted has been dermatomed to about the level where in vivo the dermal vasculature would be clearing it from the skin. And that's essentially also where the receptor solution is now interfacing with and collecting this information. This data happens to be an in vitro, in vivo correlation study with the dosing of benzoic acid, which is actually very rapidly converted to hippuric acid. And then the in vivo data is the urinary elimination of hippuric acid. This was some of the early data. But I'm not going to focus on the data. I just want to talk about the technique for now. So if you're going to compare a test and a reference product, you then, on the same donor, dose the reference product on multiple replicates. And because the variation from one individual to another can be substantial in terms of permeability of their skin, you also want to take a look at different donors in the population. Same anatomical region, but different donors. And so you do this with a whole set of donors until you have a sufficient population that's representative and adequate for your study. Now, I'm going to focus a little bit on the diffusion cell system as well. So this is a classic depiction of the Franz diffusion cell. But over time, there have been innovations, and there are currently numerous models that are available, including this one, which is the Teledyne-Hansen diffusion cell. It is also a vertical diffusion cell. It has variations. For example, this is a jacketed diffusion cell designed to have a water jacket that maintains the skin at the physiological temperature. Well, these ones are not jacketed. They use a different mechanism, and they have variations in their receptor volumes. They have variations in their donor compartments. There's automation that's available with them. There's another system by Logan that also has other innovations. For example, a design for eliminating bubbles, a corrugated flange that helps with the gripping of the skin. Also with automation, and has features like a plexiglass shield to be able to better regulate the environment that the diffusion cells are exposed to, even in labs where there may be, air conditioning or vents or other things happening. There are also other types of diffusion cells, like this flow-through diffusion cell, which, unlike the static diffusion cell, static meaning that the receptor solution stays as it is until it is exchanged at a sampling time point, whereas with these flow-through or inline diffusion cells, the receptor solution is continuously being exchanged as it flows under the skin. And this is also available with innovation. These are Permigure systems, and this is also a Permigure type diffusion cell. So we are very fortunate today, and I'm incredibly grateful to all of the manufacturers of diffusion cell systems out there. These are just a few of the very common models. There are others as well. All of these are excellent diffusion cell systems that can be used to develop excellent IVPT studies. And I'm very grateful for the investments that all the manufacturers have put into developing methods, to developing, I should say, models of diffusion cells that have made this increasingly efficient to do these studies. And they're actually here and have been participating in this workshop, which I'm very grateful for as well. So a big thank you to them. So let's move on now. We know what IVPT studies are. We know the kind of equipment that's used for it. And by the way, we use the term equipment, not apparatus, because unlike apparatus 2 or apparatus 5, that are compendial apparatus, where those precise dimensions of the apparatus are going to be consistent regardless of which manufacturer you buy it from, here diffusion cell equipment has innovations and variations that are a little bit different among them. So we call it IVPT equipment. Now, let's go back in time a little bit to understand what's the history of IVPT. So when we go back into about the 70s, we see that there was a lot of research being done to evaluate the in vivo relevance of IVPT studies, the in vitro in vivo correlation, for example, and many of these were in fact with transdermal products because it was possible to characterize the bioavailability of transdermal products more easily than topical products. So this is some data set with scopolamine. This is the data set that I just talked about a little bit recently. This is testosterone and estradiol. These are just a few of about over 90 in vitro in vivo data sets that showed very good in vitro in vivo correlation. And because of that, the question arose, could this methodology, IVPT, be useful when developing a generic product whose bioavailability rate and extent was intended to match that of the reference product? And a good story is that of ketoconazole, where in this instance, this was the reference product bioavailability. And so different formulations were evaluated by IVPT so that the formulators could identify what characteristics of the formulation needed to be modulated to better match the performance, the rate and extent for the reference product. And in fact, this qualitatively was found to be a good match, went into a clinical study and was found to be bioequivalent. So as the years went on, the qualitative assessment and utilization of IVPT in this way evolved to be more quantitative. Here we see two steroids where the test and reference creamer, this is the prospective generic product during development for creams and ointments of these two steroids, were evaluated by IVPT. And now it was starting to get a little bit more quantitative. And in fact, when they looked at, they didn't have endpoints yet per se, but when they looked at the test to reference ratio for the bioavailability, it was very, very well matched among all the test and reference products in vitro. And in fact, when these steroids were evaluated in a clinical vasoconstrictor study, they were also found to have a very good test to reference ratio, and in fact, were found to be bioequivalent. Now, one of the challenges was that in vivo approaches, other than the vasoconstrictor assay, to evaluating bioequivalence were often quite arduous. These typically involved comparative clinical endpoint bioequivalent studies that were largely not compatible with the pharmacoeconomics of the generic drug industry and were often unfeasible to conduct, quite frankly. And so one of the things that we sought to do through the GDUFA Science and Research Program was to develop more efficient approaches by which to demonstrate bioequivalence of topical products and to enhance patient access to these important types of medications. And so a different type of approach to bioequivalence was envisioned. And this is really where the story of IVPT intersects with the opportunity to be able to support demonstrations of bioequivalence for products who by design were as closely matched to the reference product as batch to batch of the reference product was to itself. And how did we know that? How did we characterize that? Well, we established a set of principles to be able to qualify these products based upon the components, where Q1 characterization provided a profile of the quantitative components, the ingredients, the qualitative components, thanks, Markham, the qualitative components in the formulation. Q2 was the quantitative composition of that product, providing a profile of the formulation composition. And Q3 was the arrangement of matter in that product, where Q3 characterization provided a profile of physical, chemical, and structural attributes that was quintessentially characteristic of that product. And the research showed us that it is through the physical, chemical, and structural mechanics, the machinery, the molecular machinery of the dosage form, that these products really modulated the bioavailability, the rate and extent of bioavailability. So the role that IVPT was able to play is that as you got into more complex dosage forms, for example, emulsions, IVPT was a biorelevant test method that could sensitively discriminate the rate and extent of topical bioavailability between formulations across a wide range of drugs and dosage forms. It provided biorelevant results that correlated with in vivo bioavailability and successfully predicted the bioequivalence of topical generic products. It supported the demonstration of bioequivalence for prospective generic products that were closely Q1, Q2, and Q3 matched to the RLD. However, while it had this potential, at the time, there were no established procedures for validation, for BE study design, or statistical analysis, and there had been no systematic assessment of interlab reproducibility with positive and negative controls for BE. And this is really where the story with acyclovir came to bear. This was a product that was notoriously unfeasible for which to develop a generic using a comparative clinical endpoint study. And so as positive and negative controls, we evaluated several of the name brand Zovirax acyclovir cream products, as well as some Austrian products that were pharmaceutically equivalent. And when we evaluated them by IVPT, we developed very nice IVPT profiles for them that were very well matched among the positive controls and also very well matched among the negative controls, but well discriminated between the two sets. Now, this is just one lab's data set. In fact, what we sought to understand was if you control the product and the dose, but wanted to understand what would be the impact of using different dosing techniques, different skin types, different skin thicknesses, different equipment, different barrier integrity tests, would you still have a methodology that gave you a consistent and accurate answer about bioavailability and bioequivalence? And in fact, in all of these different labs across the world, the methods were successfully developed and they gave a very consistent answer in terms of the relative bioavailability of positive and negative controls. And these are the studies that supported recommendations, for example, for using vertical diffusion cells as well as flow-through diffusion cells, using dermatome skin, recommendations regarding barrier testing, dosing and sampling recommendations. And I will say this is not just this data set. In fact, even in this data set, there was a tremendous amount of data. I'll actually expand this to talk. There was multiple products studied here, and I'm going to focus a little bit more on the two products here, which are Zovirax products marketed in the US and the UK. Now, if you look at the underlying data for this data set, this was a 6-donor, 6-replicate data set, you can see that there are some features of the skin and bioavailability that are quite apparent here. So these are the IVPT profiles for the study population using the replicate design with multiple donors, each with multiple replicates per treatment. And this is really the best way to discriminate meaningful differences in the rate and extent of topical bioavailability across the population. But, and this is what I want to point out here, there can be substantial differences between individuals in the magnitude of permeability and sometimes even in the shape of the permeation profile, perhaps due to genetics or diet or other factors. So if we see the top row, for example, here, for a given product, which is the same product in the top row, and then this is a different product in the second row, these two products here. What we can see is that donor 1 has a somewhat lower permeation than donor 2. And donor 2 has a somewhat different permeation profile compared to donor 3 or donor 4. Even donor 3 and donor 4 are a little bit different. If you dosed a test product on donor 1 and a reference product on donor 2, you really wouldn't be able to draw any conclusions about any differences being attributable to the donor compared to the product. So it was very important that products be compared, directly compared, test and reference product within the same donor for each donor in the population. Also, we can see that that this is a pilot study really with this is about 6 donors. And this is where the recommendation for being able to use pilot studies with about 4 to 6 donors came from, because as you can see, even in this pilot data set, even accounting for the different types of variability that we were seeing here, we were able to get good information about the discrimination between formulations. And really what I want to get across here is that the recommendations in the guidance have been based upon extensive research with numerous drugs and dosage forms in academic, industrial, and FDA labs. And I'm only showing a few examples here that illustrate the nature of IVPT study results and how IVPT came to be developed. But we're going to go into this a little bit more through the course of the workshop. Now, the question becomes, great, this qualitatively is very helpful, but what is the most efficient way to conduct an IVPT study, a pivotal IVPT bioequivalent study that has sufficient statistical power to be able to demonstrate the bioequivalence of a prospective generic product. Like, how many donors and replicates would we need? And this is something that we evaluated with these data sets. So staying with this particular graph right here, comparing these two products, the Zovirax product marketed in the US and a product from a different container closure system marketed in the UK, that accounted for some of the difference in bioavailability here. What we see here is we know that the variability inherent in human skin reflects real differences in clinical bioavailability. So we don't want to get rid of the variability. We're not trying to control that. That's meaningful variability. What we're looking for is a statistical evaluation and analysis that can help us draw conclusions despite that variability. And when we evaluated the approaches, there were two that were conceived. Well, people thought, IVRT has broader acceptance equivalence limits. So rather than the 80 to 120 bioequivalence limits, they were looking at 80 to 120 with 75 to 133. So would that be the appropriate measure here, that we broaden the bioequivalence limits? Or could we use a reference scaled approach based upon the fact that this was a replicate study design? We evaluated this. And so when we looked at the number of donors that it would take to evaluate a conclusion about bioequivalence, to power at 80% power, a conclusion about bioequivalence between these products, if we just broadened the limits to 75 to 133, it would have still taken over 36 donors. If, on the other hand, this is at 80 to 120, it would have taken over 36 donors. If we broaden it to 75 to 133, it would have been around 22 donors. But the power of using a reference scaled analysis is that this now brought it down to about 14 donors. In fact, when we looked at this across numerous products, we were able to conclude that typically with 1 or 2 dozen donors in a multi-replicate study design, that we should be able to power conclusions about bioequivalence with IVPT studies. And this was sort of relatively similar in scale to what one might expect for an in vivo plasma PK study. And so this is really where the focus on that, the recommendation for that reference scaled average bioequivalence analysis came from. And when we used this analysis to verify, in fact, would it accurately confirm products to be bioequivalent to themselves, and would this methodology, this statistical analysis, sensitively discriminate non-bioequivalent products to be different, we found that the method would be accurate, sensitive, and reproducible using these approaches. We also looked at the number of replicates and found here that the increase in replicates from 2 to 3 increased the power, and going from 3 to 4 also increased the power, with the added advantage of weighting the mean beyond just having 1 low, 1 mid, 1 high, but that 5 or more replicates were generally expected to provide only marginal increases in the power of the study. And again, this is where our recommendation for roughly 4 replicates or no less than 4 replicates per donor came from. And when we looked at positive and negative controls, one of the things we did to get positive controls was to compare a product to itself. This is 6 donors and 6 replicates, and we split it into 3 and 3 replicates each, as if it had been parallel 6 donor, 3 replicate studies, to see if that would power a study that would prove itself to be bioequivalent to itself. And in fact, it did. And the products were found to be bioequivalent to themselves in this instance, and in fact, for all of the products that we evaluated the positive controls on. And similarly, for negative controls, where the products were determined not to be bioequivalent in vivo, these same products were studied in vivo and also found to be not bioequivalent in vivo. Now, this is just the acyclovir data set, and I won't be able to go into so much detail with all of the data sets we looked at. We did very similar studies with lidocaine and with prilocaine, where you could see here that positive and negative controls were found to be kind of selective. The method was selective to identify differences between them. And we also started to look at methodological validations to see whether sensitivity could be evaluated with different dose amounts, in this case, looking at 5, 10, and 15 mg per centimeter squared. But it happened to provide some discrimination here at the upper end, with the main concern being that if you dose the product at too high a dose, then for the same reason that it won't, just just doubling the dose applied is not going to double the bioavailability. It's not proportional in that way. If you sort of maxed it out, it may not be able to differentiate differences between products. So especially on the high end there, you want to be able to show that the methodology is able to discriminate higher differences in bioavailability. In this case, it was able to show it, but we also learned that this may be too close, to go only, let's say 50% higher and 50% lower may not be sufficient precisely because bioavailability is not proportional. And you might have to go 3 times higher or 3 times lower. As we did this with prilocaine, we got very similar data. And this is metronidazole, where we also had, very similar studies. These are all where IVPT studies were very well developed with all of these drugs and dosage forms. In this case, what we started to evaluate was the drying rate curves, where based upon the metamorphosis of the dosage form, if the quantities applied, and here we're looking at the target dose of 10 milligrams per centimeter squared also, but the lower dose was now 3 and the higher dose was 30 to be able to really maximize the difference between the drying rate profiles so that we could see that as there was more differentiation in the metamorphosis of the product, you would get a control of differentiation in the bioavailability as well. And we also looked at the evaluation here of using a vertical diffusion cell versus a flow-through diffusion cell with different flow rates. So we might come back to this when we talk about method transfer later in the study. To keep going then, where does that put us? What is the state of the field and of topical bioequivalent standards? Well, IVPT has been central to facilitating the establishment of modern bioequivalent studies. In addition to publishing and presenting the results from IVPT studies extensively, we've utilized these new insights to establish novel and efficient bioequivalence pathways and modernize regulatory standards so that in these guidances, the Q3 guidance and the IVPT guidance, and other guidances, we now have a framework where if the arrangement of matter is the same for the drug product, and if the underlying matter is the same, then in many of the situations, an IVPT study is not even needed, but for more complex emulsions and all, it becomes an essential piece. So, a portion of these Q3 same products benefit, and really we're able to spread the entire range of them by having IVPT studies in those areas when needed. And these are also very valuable. IVPT studies are very valuable in situations where there may be differences in the Q1 and Q2, but the Q3 is preserved, and IVPT is going to play an important role for these Q3 similar products as well. Now, in our recommendations, our guidances for industry and our PSGs describe bioequivalence approaches now for almost all topical drugs. And many of these, the characterization-based option is made entirely possible because of an IVPT study. And so as we recommend these studies, we've developed the methods, we've shown the feasibility for these with in-house research at FDA. This is, for example, just from very recent PSGs on roflumilast and clascaterone. And we put these out in posters at scientific meetings, in publications. And this has really allowed us to have quite a bit of success for the American public. This has meant that efficient in vitro characterization-based options are now the dominant approach for topical ANDAs submitted to FDA. In fact, more topical generic drugs have been approved in the last decade alone than in all previous decades combined. And several of these approved applications were supported by IVPT studies, in fact, well into the double digits. Yet topical products are complex, and IVPT studies can be challenging to develop, to validate, and to analyze. And so this workshop will illustrate and discuss some practical ways to address these challenges. And we're going to do this with multiple diffusion cell systems. And I'm so excited to be able to be here over the next couple of days with all of you. I do want to acknowledge that this has been an enormous number of people around the world who for many years have worked very hard, to bring us to the point that we are today. Among all of them, I'm acknowledging some people who specifically are involved with the work that was on my slides here, but I want to in particular recognize Dr. Priyanka Ghosh, who from the very beginning of this journey has been at the center of every study, every design, every regulatory concept, every workshop that we've held. She has been the heart and soul of the program and continues to lead, and her team, the program. And we're very, very fortunate. So a very special thanks to all involved, including the many research collaborators, and of course to the Center for Research on Complex Generics, who's hosting this workshop today. Thank you so much, everyone.
Okay. Okay. I don't see the presentation on the screen in front of me. Oh. Okay, let's do this the old way. So thank you so much, Sam, and to everyone. Like Sam said, there was a huge group of people that was involved in both planning the workshop and also also assessing all of the ANDAs we have received to date. So what I'm going to do now is talk a little bit about, now that we know how the guidance was developed, what ANDAs have we received, and what are some of the challenges that we have observed in these ANDAs. Oops. Okay, a brief overview of my presentation. I'm going to talk about what product-specific guidances we currently recommend IVPT in. I'm going to also then talk about what ANDAs have we received and what are some of the challenges we have observed and what are some of the opportunities that may present for all of us to learn about next steps. So within the scope of our overall BE landscape of PSG recommendations for topical products, we historically, we have recommended these comparative clinical endpoint studies. These studies can be used by all topical products, irrespective of the formulation. We have recommended vasoconstrictor studies, which are primarily used by corticosteroids. We have these options for waiver of in vivo BE study requirements, which can typically be used by simple solutions where bioavailability is self-relevant. And then ultimately, we have the characterization-based approaches, which Sam referred to in his talk, where we look at, these are mostly applicable to products where the formulation is extremely well matched to the reference standard. Then we have the Q3 characterization, we have the IVRT studies, and then in some instances, we have the IVPT studies. Since we already heard about the instances where we do recommend these IVPT studies, sorry. Okay, I don't know how to go back, so we are going to go with this. That's fine. Can we go to the next slide, please? Thank you. So at the moment, we recommend IVPT studies in about 31 guidances, and they are mostly for the creams, the ointments, or the lotions, the emulsions, where we really need to understand. Perfect. Oh, okay. So we recommend it for the emulsions, primarily where we really need to have an understanding of the interaction of the prospective generic product and the reference standard on skin to understand if differences in manufacturing or differences in, say, for example, the grade of the inactive ingredients may influence bioavailability to support an overall evaluation of bioequivalence. So with these 31 PSGs that we currently have out there, we have received a lot of ANDAs. So to break down the ANDAs that we have received so far, I'm going to start with all of the topical ANDAs that we have received since FY 2018, which is September of 2017. We have received about 350 ANDAs in total. And for the purposes of this presentation, all ANDAs for a given year are normalized to the total number of ANDAs in that year. Approved ANDAs for the purposes of this presentation are all the ANDAs that are either approved or tentatively approved. And then pending ANDAs, which are shown here in green, are all ANDAs that are pending either with us, the agency, or with the applicant in a CR status. So overall picture that we see from this graph here is we have about 60% of the ANDAs, give or take, are already approved, about 200 ANDAs in total. And then the pending ANDAs are lastly for the last few years, are primarily for the last few years, which are still undergoing the review process. Now, if we take a closer look at the ANDAs that have used the characterization-based BE approach to support their submissions, we see that over the last few years, there has been a sharp uptake in using these characterization-based approaches to submit these ANDAs. And of these ANDAs, a lot of them have been approved. So we are talking about, give or take, about 80 products that have been approved using these characterization-based approaches. But what I really wanted to point out on this slide is in these ANDAs that have been approved, IVRT studies have been successfully completed for a lot of these ANDAs. And also, like Sam mentioned in his talk, IVPT studies have also been successfully completed in a lot of these ANDAs to support product approval. So while we acknowledge and appreciate that there are a lot of outstanding challenges related to IVPT, we did want to note that IVPT studies have been successfully completed to support products that are currently on the market before we delve down into the challenges a little bit more. So now let's talk about the pending ANDAs and where our challenges lie. I'm going to take a step back here and talk a little bit about overall all of the pending ANDAs that we have. And we see that in about a little bit more than 50% of those cases, the remaining challenges appear to be related to either pharmaceutical quality, which is related to, again, drug substance, drug product, or the manufacturing, and or related to the labeling. And for the other about 45% or so, we have challenges either related to bioequivalence or pharmaceutical quality and others, which include labeling in certain instances. I wanted to take this step back because I think when we are interacting in the pre-NDA space, it is possible that there are some questions that would be helpful to ask or would be helpful to discuss related to pharmaceutical quality and others that in the future could help to move these ANDAs forward as well. Now, delving more into the BE-related challenges and specifically focusing on the characterization-based BE approaches, if we look at the pending ANDAs that we have for characterization-based approach, we see that in blue here, there are ANDAs where the IVRT study is already adequate, but the ANDA is still pending, maybe related to pharmaceutical quality or other issues. And then we also see a group of products where the IVRT studies are still pending. Now, while there seems to be about, say, 40% of the products there where IVRT studies are still inadequate, I do want to note that a lot of the products are still under, the first cycle review. And also, in a lot of cases, some of these challenges are easily resolvable. Like there are questions related to missing information related to the formulation that was used in these studies or to the manufacturing process for these formulations, or small questions related to method validation, which are often resolvable within the DRL stage or within the first cycle stage. And the main reason I wanted to mention it here is if we are able to get to a point based on the interactions we have, based on our guidances and recommendations that are provided in workshops like these, we may be able to get these ANDAs to the first cycle approval in the future, which only contain like Q3 and IVRT studies. For IVPT studies, the problem seems to be a little bit deeper at the moment. So in the pending ANDAs, we see only a very small subset of cases where the IVPT studies are already adequate. There are a lot of instances where the IVPT studies are inadequate at the moment. And on its face, it appears that the primary challenge with the IVPT studies are related to IVPT method validation and specifically related to establishing the discriminatory ability of these IVPT methods. However, and there are a few instances, of course, if the method is not validated, we cannot conclude that the pivotal study is adequate. So there are challenges related to the pivotal study at the moment as well. But if we take a deeper look, we really begin to understand where the problems may be. So when we look at the pre-NDAs and the ANDAs that we have with us, what we really begin to see is there appears to be either inadequate method development or that the method development document that we have access to, that we are reading, does not adequately explain the thinking behind the method development process. For example, we often find that a receptor solution with a significant amount of a surfactant was chosen for a given product. But there is lack of information in the method development report about how that receptor solution was systematically chosen. And how do we know that that is an appropriate receptor solution, has adequate stability, adequate solubility to support that IVPT method? Similarly, we often see examples where the sampling duration or the sampling frequency have not been optimized, and we see a very standard 48-hour study with 4-hour sampling frequency, and I'll show you the impact of that on the next slide. So what we have elected to do in this workshop is spend almost the rest of today talking about how to develop an IVPT method, and more importantly, how do we determine that an IVPT method has been adequately developed so that that method can be moved into method validation and pivotal study. We are, of course, not going to be able to address product-specific questions within the workshop, and we do encourage you to interact with us through the product development meeting pathway or the control correspondence pathway when you are developing an IVPT method and you run into challenges and you have questions related to method development issues or even method validations issues for that matter. The second big example, like I said, is related to method validation. And here the main problem we see is related to assessing if a method is sufficiently sensitive and selective. And some of it comes from the selection of the dose, like the example that Sam gave. Some of it comes from the fact that there is no quantitative methodology at the moment that is used to assess sensitivity and selectivity of the method. So we are going to spend entire morning tomorrow talking about what are FDA's expectations for assessing sensitivity and selectivity, and what are some of the deficiencies we are seeing in the applications. And again, for more product-specific questions, we strongly encourage you to engage with us in product development meetings, even pre-submissions meetings when appropriate, so that we can resolve these challenges prior to the ANDA being submitted. The last thing I'm going to cover today is aberrant data. So aberrant data is something we discussed in detail in the 2021 workshop that Dr. Polli referenced in his opening remarks. And some of the considerations that we heard related to aberrant data in this workshop is we are already seeing data sets that are so very variable, and by allowing, for example, statistical methodologies to identify outliers or removing aberrant data, are we basically going to remove 50% of the data set? Or are we going to basically allow quote-unquote data cleansing before we use a data set for BE analysis? So I'm going to set the stage by saying when I talk about aberrant data, I'm talking about a situation where this is a real example of a study where 5 replicates for the test product and we have 5 replicates for the reference product. And one of those replicates in a complete data set appear to behave very differently than the other 4 replicates for the same product within the same donor. And we just heard from Sam about the importance of evaluating the test and the reference product in the same donor at the same time to really understand the performance of the product. Now, I do want to mention something here. This study design, looking at just this one donor data, may not appear to be ideal. Of course, it doesn't seem like it explains, it is able to capture the metamorphosis of the product in its entirety within this 48-hour study. The sampling intervals also don't seem to be ideal. But the real, I did want to pick a real case example to show that even in data sets like this, we often see that there are skin sections where the product behaves completely differently than the other 4 replicate skin sections for the same product. So in our study, we looked at about 41 data sets, and we tried to really identify how often does this happen when we use these well-known statistical tests like the Dean and Dixon test and the Grubbs test. And we use different alpha values, mostly to look at what is the impact of alpha value on identifying these outliers. So the first thing we noticed is When we used an alpha value of 0.05, in about 95% of the cases, we identified an outlier. So of these 41 studies, we identified an outlier in like 40 of them. The second thing that we identified is the outlier percentage is not like 50 because, of course, we put restrictions on the test. And the poster is hanging somewhere in the adjacent room, I believe. So you can look at the steps we took here. But the outlier percentage that we observed in this data set of 41 pivotal IVPT studies is around 2.3%. So we are talking about 3 out of 120 cells that were identified to be outliers, or 10 out of a 360 cell data set, which were identified to be outliers. And the last thing that we observe is what is the impact on an assessment of BE. So we assess the data set per the recommendations in a guidance where we don't remove any data, and then we assess the data set after removing the outliers. And we saw that 2 out of these 41 data sets only pass after we remove the outliers. 7 out of these 41 data sets fail after we remove these outliers. But I would like to point out that this is something that we are doing internally as part of our research to really follow up on your recommendations from the last workshop that there is aberrant data in these IVPT data sets. Our current recommendations, our current guidance indicates that in the pivotal studies, we should not remove aberrant data unless there is an assignable cause to that specific cell or to that specific specific apparatus and equipment as such. However, what we would really like to hear from you is what are you observing in your IVPT studies, and what would be your recommendations for how we handle outliers in these IVPT data sets, understanding that in some instances, what gives us the best understanding of the metamorphosis of the dosage form and a comparison between the test and the reference products in these data sets. So with that, I would like to conclude by saying that we looked at all of the ANDAs that we have received since FY18, and characterization-based approaches have been predominantly used. We have of the 350 ANDAs that we have received, we have already approved about 200 of them and 80 using characterization-based approaches. But there are challenges, and specifically challenges related to the ANDAs that include IVPT studies, which mostly result from appear to mostly result from inadequate method development and related to the method validation study. And like I said before, we strongly encourage early engagement with the agency through the PDEV meetings or PSAV meetings to resolve some of these issues prior to submission of these ANDA. I'd like to end my talk by acknowledging our fantastic ORISE fellows, Ling Xiaoxi, who worked on collecting all of the data related to the ANDA projects, and Jackson Russo, who is in the audience today and really led our efforts related to the aberrant IVPT projects, as well as our entire team within the Office of Generic Drugs, who has supported the IVPT program over the last 10 years or so. Thank you so much for your attention.
Our third speaker is Dr. Lakshmi Raghavan. He's a successful serial entrepreneur with more than 25 years of technical and business development experience. He is currently the founder and CEO of Helios Labs, LLC, and he's leading its overall corporate strategy and business development and R&D. Prior to this, Dr. Raghavan founded Solaris Pharma Corporation in 2015, which is a company dedicated to developing generic and branded pharmaceutical products. He also founded Dermpath Pharmaceuticals in 2010, which is a contract research organization. Dr. Raghavan holds a Master of Science in Physics and a PhD degree in Physics from Indian University of Technology from India. He has more than 40 research publications and 8 book chapters.
Thank you all for inviting me for this workshop.
Don't click on the mouse.
Okay, so for changing the... okay. Thank you all for inviting me for this workshop, and especially to convey some of my thoughts around the IVPT studies. We at at Solaris Pharma Corporation, we were one of the first to adopt the in vitro guidances, a lot of approvals on the IVRT, and significant experience with the IVPT. So I would like to share some of my thoughts on this. I founded Solaris Pharma in 2015. And when I founded that company, the IVPT guidance probably had just come out. And we had several products in pipeline that required a clinical endpoint study. And so this came as a blessing. And from one product where we could do a clinical endpoint study, we had 3 or 4 products that we could put with the in vitro guidances. So that becomes an affordable option, especially for a small company like ours. And taking the acyclovir guidance at that time, we started developing IVPT product. And soon we realized that there is going to be significant challenges in adopting them. So we went through those exercises, and then we had a lot of interactions within the industry and with the FDA. And again, it was another blessing that we got another guidance which had a lot of details which we didn't know at the time when we started about how to conduct IVPT studies, especially starting from method development, validation, and execution. So I'm going to share some of my thoughts on some of the challenges that we have faced, and also see how we can resolve some of those challenges as well. So the title, as it suggests, it talks about challenges, but also I would like to point out some of the things that we can take care of while we are conducting these studies. Outliers, we all have heard about this. The FDA has spoken quite a lot on these in earlier conferences, and even here today we have heard about it, and what type of outliers we can expect, and can we even address those statistical outliers, which seem to be a major challenge. So again, for those who are starting up IVPT studies, some of the challenges that you will see is even the setup costs are pretty expensive. You have to buy several pieces of diffusion setup. You need to have several, mass spectrometers, especially if you want to scale up and do several studies at the same time. The study costs themselves, I've spoken about this in previous meetings, that it is relatively cheaper than a clinical endpoint study, but for those who are starting up, it could still cost quite a lot, especially if you are outsourcing this to another lab. The time development validation, it can take anywhere from 24 to 32 weeks, depending upon the type of equipment you have, number of equipment you have. And again, IVPT studies are relatively complex. And as you saw, statistically, more IVRT studies have been approved compared to IVPT studies, which we are all working together to see how we can come up with some of these strategies to have more IVPT approvals. I'll be talking about some of the inherent skin variability that we see, what are the experimental variabilities that you can address. And if it is a product variability, of course, it's going to fail because it's not going to be bioequivalent. And how to differentiate between experimental and statistical outliers. And also, we need to look at approaches to address these outliers. Again, just to give you an idea, France type diffusion equipment, we saw different manufacturers of these setup. There are many of them use static vertical diffusion system, but there could be occasions where you would need flow through cells as well, especially where the permeation is high. And if you want to scale up and do more products, you would need number of diffusion system and also number of mass specs, which can run into several hundreds of thousands of dollars to even set up. In terms of timeline, these are typical. This can be lower if the method is easier to develop, relatively easier to develop, and it could take longer time as well. We have taken longer time to develop one of the IVPT studies. So it just gives a rough idea, as I said, about 8 months, 6 to 8 months to do this study. And the total study costs, I already mentioned that it can be even 500 to 600K, depending upon if you're outsourcing it. And one of the challenges that we have seen also I want to express here is you take about 6 to 8 months to do the study, you submit, get a response, and especially if you're getting a CRL, for example, your RLD has expired, how do we handle this type of a situation? So I just want to talk a little about the types of outliers that we can see. There are 3 different types in my mind. So there are experimental outliers, or I would say experimental variables that you can get while you are conducting the experiment. That could be statistical outliers. We have seen some of the data earlier. I will also show some of the data we have seen from our experience. It could be intra-donor, or we have even seen some inter-donor variability. And as I mentioned, if the outliers are due to product variabilities arising out of the product itself, there is a good chance that your IVPT-BE study will fail. So what are some of the experimental outliers? There could be 2 different ways, identifiable. The identifiable outliers, like these are what you observe when you conduct your experiment. You can get these type of outlier before even you start your study, like the TEWL may fail from your acceptance criteria. It could be inhomogeneous application that you observed. Some of the drug product gets stuck to the side of the cell, or skin is damaged. You observe that it is damaged during application. If it's during the experiment, there could be a skin slippage that you can observe. There could be bubbles in the receiver phase, not mentioned here, but there could be an equipment failure or cell temperature that you monitor periodically during the course of the IVPT study. It is fluctuating and it goes outside of the limit, or even sometimes, the room temperature and humidity can be different to what is normal accepted range. And after the run, you do your analysis, you see some identifiable contamination from either your previous cell or from your experiment, or other analytical errors like you have a vial missing or so. Then you provide a rational justification for identifiable causes, and then you can replace the cell in that case, or donor. There could be other unidentifiable causes. We have seen some ghost peaks in the chromatograms where you have not seen it for any of the other replicates or other donors. There could be some unknown contamination that you see in the chromatogram not arising from the drug itself, or you have a detection of drug in T0, which means you have not cleaned your cells from the previous run. So, again, for any of this to be a valid reason, you need to have a good rationale and try to identify it before you start your analysis as much as possible. And, and what you would also do is put it into your protocol as much details as possible, so that, you can look at these outliers when you're doing the data analysis. Outliers arising out of variability. It could be intra-donor variability, variability in replicates within the donor, or one or more than one cell showing unusually high or even low permeation. Or it could be, I'm going to show you an example of where you can get some inter-donor variability, or one or more donors, again, showing unusually different profile than the rest of the donors. The key question is whether the variability is seen in both the RLD and test product, or it just happens in either test or reference product. The intra-donor variability, this is an example where we saw one example from Priyanka as well, that you have here 4 cells that are giving a very similar data, and one cell giving unusually high value, almost like 5 to 6 times higher than what you got for the rest of the replicates. So this clearly shows that, the drug product itself behaves well. And you see that in the initial time point, the data is similar, and then it deviates away from rest of the flock. This gives another example where, there are 3 different donors, and each donor, when you do, on the left-hand side, we saw on the previous slide, in the middle, you see that you get a nice profile, and then, in the last one, you see that the Tmax, the Cmax occurs at early time point. So it's the same product, same method, and different donors, you still get these type of variable behavior. So I'm just giving another example here of what we have seen in interdonor variability is you see that the data is very consistent on the left-hand side for donor 1. As you will see in the flux data, which shows more clearly, you will get a nice profile. And then on the right-hand side, suddenly the whole profile moved to a much later time point, and you see very low permeation to almost 0 permeation until about 48 hours, and then it starts off at 52 hours. And you see it just starts to take off after that. If I just show the same flux data, you can see that this is test and reference here. And you see that the profile is very clearly shows a peak, and then it comes down in donor 1. And donor 2, you don't see the Cmax here. This was really surprising for us. And you see that the lag times are different. And what you see here is unusual. But when you get this data, that the Cmax and the AUC can be totally different to what you would expect, because you're still on the lag time phase and the increasing phase in donor 2. So in the same study, this is again another example from the pilot study data that you see here. We came out with a statistical number. In order for us to be conservative, we did about 24 skin donors in this study, which is quite a lot, and especially considering that you see most of the Cmax around 48 to 52 hours. You needed to run the study till about 84 hours in this case. So it's a very long study that we get. So this shows the actual T/R ratio. You can see that, of these 24 cells that you see here, there are 2 that are in red that gives a totally different T/R ratio. And that came out because these 2 donors had a much later permeation starting after 52 hours. So you don't see the actual Cmax in these cases. If you remove those 2 outliers, you would see that the study passes. But one thing I wanted to point out from the previous slide was that, that behavior, unusual behavior happened both for test and for the reference product, indicating that, it happens in, it's not a product related, it is more a skin related outcome in that case. So how can we address these outliers? I suggest two of the methodologies that one could adopt. Having said that, I have not tested any of these hypotheses. This is just a proposition. So when you complete your pilot study data, you normally come out with a statistical analysis and say that you need certain number of donors. Now, what you also can do is use a non-parametric approach to rank order the pilot study data for both AUC and Cmax. Let's say I just take an example, it's random, but it could be any other number, but you need a minimum number of data points to go through this approach. Let's say you have 10 time points with 5 donors and 5 replicates, test for $D. you do the ranking order and then you define outliers based on the ranking order. For example, it could be 3 times. This is just a random number again. And then, you incorporate the outlier criteria in the pilot study protocol. Once you complete the pilot study, identify the outliers and you can replace the cells. And then, you include the data in the statistical analysis. So that is one approach. So this just shows the same in a flow chart. From bottom up, you provide the same pilot study data. You know the number of donors you want to go for, and then you conduct the non-parametric analysis, and then add that into the protocol before you conduct the study. And then once you conduct the study, repeat the cells that are outliers, and then you can add it in the statistical analysis. So this is one approach. The second approach is more like an adaptive tiered approach where, let's say you have 14 donors you're starting off with, you can, define a smaller number where you will do an interim analysis of the statistics. And then, data from this, you can see these are predefined rules that you have established in the study protocol. And then you have clear guidelines of what you would do in the interim analysis. This again would be from the experience of the pilot study. So you can do a sample size re-estimation at that point, and then see whether you would need to increase the number of donors. If so, by how much? Or if you have passed the study, you would be able to conduct the statistical analysis at this point. Again, these are very initial thought process. A lot of analysis needs to go into it, test these hypotheses, and hopefully, there will be a solution to it. So again, the same thing in a flow chart, just to make visual easier to understand, but it just says the same thing as what I just described. So overall, the key to successful IVPT studies is to overcome the challenges. Of course, we all go through these experiences, but I think we have come a long way from where we were about 10 years ago. Now we have a lot more understanding and a little bit more push to overcome some of the challenges I mentioned here would go a long way in helping us as industry to perform more IVPT studies for regulatory approval. One thing I would like to point out is you need significant knowledge and expertise. Just conducting the IVPT study alone is not a criterion, but you should understand what the data mean. How do you interpret the data? How do you differentiate good and bad data? How do you conduct this study efficiently? So all of those are key to a successful outcome. And I'm hoping that, as an outcome of this workshop or in future, we will have alternate approaches to addressing some of these statistical outlier where we identify the problem as due to the skin and not due to an experimental error. So this is my contact information. If outside of the workshop, if you'd like to reach out to me for any questions, please do so. Thank you.
So our last speaker in this session is Dr. Aaditya Bhatt. He received his master's and PhD degrees from St. John's University in New York in the field of medicinal and pharmaceutical chemistry. He joined Amneal Pharmaceuticals in 2015 and served various roles, which included pre-formulation development, support characterization studies, and BE studies of complex dosage forms. And his roles and responsibilities include overseeing in vitro development programs for complex dosage forms and amneal. In the past few years, he has been actively involved in overseeing the development and execution activities of IVPT studies for topical and transdermal dosage forms.
First of all, thank you. Really thanks to FDA and CRCG to conduct this workshop, which will give a vision and path forward for conducting the IVPT studies next course of action. We at AMNIL have been working on IVPT since more than 10 years. And over the time, we have gained experience, knowledge with more and more data for different products, and also collaboration with the agency through different mediums to get an idea and insight on how to conduct IVPT studies. And as they say that as we do more, as we work more over in the field, we'll get more knowledge into it, and that's how it matured for us. So moving on to before starting the IVPT study, the challenges, based on our experience, we would like to briefly go over the IVPT conduct. So this is an overview. Of course, it is subjects to change for specific different products as we move through. The IVPT method development, it's a crux of the IVPT subject. So it involves several things like skin selection, equipment selection, as rightly mentioned by the fellow speakers. Skin selections involves there are different sites of anatomical sites of the skin. So we evaluate the products on different sites and we identify which is the suitable sections and the anatomical site which are suitable to assess the IVPT study for the samples. For the products, equipment selection, as we know, there are known instruments like Logan, Hansen, flow-through cells. So these are the different equipments we evaluate during the course of IVPT study, the method development. Sampling, which involves the sample receptors media collection. We evaluate the different sampling regimen to identify which is the appropriate sampling regimen to have a well-resolved flux profile as the requirement, which is the critical requirement of the IVPT, which involves rising phase and the subsequent lag time. I mean, the decreasing, I mean, the lowering of the flux, the dosing duration, application of the dose onto the skin sections. Based on the product physiology, metamorphosis of the product, we select the appropriate dosing duration and media and the dose selection. The media is very important, as we all know, which the media should be inert to the analyte. Appropriate permeation enhancers are being selected to are evaluated to identify which is the appropriate permeation enhancer in case of the very hydrophobic drug, which is having low permeability capacity, and dose selection. So this all program is over more than 8 to 10 months, and in certain cases, if based on the product complexity of the product, it is more than 12 months. We go through the hardcore exercise for IVPT development, and eventually it further moves into the design of the pilot study. Based on the variability we encounter during the development program, we design a proper pilot study with appropriate number of donors based on the data set we have encountered during the development. And eventually, we design the pilot study during the validation stages. Also, the dose selection evaluation gives us an estimate of to design the sensitivity study for appropriate dose which can be selected. Also, the control of the study procedures, which are not only limited to validation, but also to during the development, because it will give an idea of how study procedures are controlled, and bioanalytical method validation, the analytical method to quantify the analyte is being validated during the IVPT validation stages. And eventually, once this entire exercise is done, the method is formalized and we move into the pivotal study. So the estimation of the pilot study based on the statistical analysis, we encounter the variability. We design the appropriate number of donors which can be selected for the IVPT pivotal study. So that's how we go forward conventionally during the IVPT study conduct. So this is what the IVPT conduct is. Now, the main topic as we are here is the, sorry, challenges of the IVPT. So the first topic which I would like to cover briefly is understanding the IVPT controls during the study. That is how the correlation of the IVPT control with the aberrant permeation profile. So as we have discussed before by the speakers, we exercise several IVPT controls to ensure that the method is being controlled to ensure that we minimize the variability. However, we still encounter the variability. And what are the various factors we try to control during the IVPT study? And still we encounter the aberrant permeation profile. So is there a way we can identify any additional controls during the experiment apart from the existing controls? And second is the skin handling of the dermatome skin during the IVPT setup, that is washing and equilibration step. So apart from during the development stages, we also try to evaluate if there is a possibility we can modify or optimize the skin treatment step during the IVPT sample, the study setup. And the last topic, which I think most of us will be discussing anticipated, is the outliers, which I will briefly go over during my presentation. So I will just explain based on the data set as to keep it short. And so this is a typical data set which we do a multi-replicate study. So typically we identify the IVPT controls, the skin thickness, the skin selected, the 12 values, that is the barrier integrity. So beginning 12 value, that is we measure prior to dosing, and the middle and end are just for information purposes in case we encounter any kind of aberrancy during the course of IVPT study. We use this information to identify if there was any kind of incident that might have happened. So these are typically measured during the middle and prior to the end. Like let's say it's a 96-hour study. So middle is measured at during 48 and end is prior to, let's say, 94 or 96 hours. Similar, the skin temperature beginning, middle, and end, the dose applied. So typically, if you see the IVPT study is stringently controlled, everything is well controlled during the entire study. However, if you see replicate one of the study demonstrate an aberrant permeation profile. If you see the flux, it's almost 1,000 times more from the rest of the data set. So while measuring this, if you see the replicate one demonstrated significantly tight numbers, but when it is reflected in terms of the data, if it's non-trending. So the question which arises here is that what aspects additionally can be evaluated to avoid such permeation results? Yes, one reason is whatever skin section was used might have been anatomically, there might be some anatomical difference which cannot be visualized by eyes. Application of the dose onto the skin, however, was stringently controlled. Still apart, we identify such outliers. So is there a tool, which we can evaluate apart from that, apart from the existing controls? So this is addressing these outliers. We conventionally say that might be an aberrant skin section which we would have encountered. But if there is a way we can come up with while conducting the IVPT study, that can help us to navigate through these challenges. Next aspect is skin handling of the dermatome skin during the IVPT setup, that is the skin equilibration step. So I will represent two donor data set where we have done one exercise where a donor was selected and IVPT study was conducted. Just the only difference was the skin treatment step. So if you see over here, the skin treatment with immediate thawing, where in the skin section, the dermatome skin pack, which was procured, was taken. It was thawed for like 5 to 10 minutes, washed, and skin was removed, quickly washed to remove any kind of adherent tissues or blood, if at all it, and eventually the skin was cut, and experiment was started by placing the skin on the vertical France diffusion cell. Skin treatment with 60-minute equilibration, where 60 minutes equilibration represents treating the skin in the media, the proposed diffusion media, for 60 minutes. And once the equilibration was done, the skin was then punched and experiment was started by loading the diffusion as we generally do. And similarly, 120 minutes represents the 120-minute equilibration time. When the study was performed, the IVPT controls were monitored and everything was intact, which represents that there was no abnormal experimental error, visual error that was encountered during the study. Moving on to the data associated with that, if you can see the skin treatment with 120 minutes, skin treatment with 60 minutes, and skin treatment with immediate thawing, all the data sets represent close permeation profiles for the respective donor. So hydration step of the skin it was observed that we could not observe any kind of aberrancy in terms of the barrier integrity results. And accordingly, the permeation was reflected in similar magnitude for, as you can see, for the flux and the cumulative permeation results. So if you can see over here, all the three treatments, 120 minutes, 60-minute equilibration, and the skin treatment with immediate thawing demonstrated similar permeation results for the respective analyte. So note that everything was stringently monitored throughout the study, and multi-donor study was done. So I will be just representing another similar experimental data where equilibration was performed 60 minutes, 120 minutes, and immediate thawing. And the data associated with that, the permeation results were slightly low as compared to donor 1, but however, we observed similar permeation profile. So as you can see over here, the data, all the three treatments demonstrate the flux profile was somewhat closer. The permeation, magnitude of permeation, though, was not significantly high, but permeation was observed and the data trend was similar for the three treatments, which brings us to a question. Can we optimize the skin handling step for the dermatome skin during the IVPT study? If these controls are stringently monitored and we have a reproducible flux profile with multiple skin treatments, if there is a way we can optimize the skin treatment during the study, while with adequate scientific justification, with an adequate data-driven justification, if that can help to carve the path for reproducible IVPT profile, that is a topic to ponder upon while we are doing the IVPT study. If there is because the guidance currently mentions the skin treatment, thawing steps, and subsequently you can perform the IVPT study to ensure that the skin hydration is not impacted. So if all these steps are stringently controlled and they are reflected appropriately in terms of the data, if we can navigate the path for skin permeation, for skin treatment during the permeation steps during the IVPT experiment. So that is another kind of a challenge and topic to think over. Of course, we conventionally follow the agency's recommended skin treatment steps, but this is just a next step if we can navigate and think upon during the anticipated studies. Third topic I'm just briefly going over is the estimation of outliers in the data set. So as you can see over here, IVPT experiment was performed on 6 replicates. This is just one representative data, but we have several datas which we encountered during method development and validations, and also, of course, the pivotal studies. So currently, agency's recommendation is not to eliminate any outliers which are not experimental, and we do follow that. However, as my colleagues have, the August speakers have mentioned, several of the approaches like the Grubbs and the Dean and Dixons, which are the statistical, we can predefine it in the study protocol during the method validation or the pivotal study. So this data set represents out of 6, two of the replicates showed the aberrant permeation profile. If you can see over here, 3500 and 260. So what we want to ask here, what we've encountered conventionally during the IVPT study is, let's say 2 out of the 6 replicates are showing aberrant permeation profile. While applying the Dean and Dixon's outlier test, we can eliminate 1 outlier out of the 6, which is significantly non-trending. What if there is an existing second outlier, which is having a very high permeation, but that cannot be eliminated with the conventional Dean and Dixon? Neither it's a experimental outlier because we did not see any kind of aberrant permeation profile. The 12 values and the dose application and everything was tightly monitored. The skin temperature was tightly controlled, which represents, it's just the very initial stage that everything was tightly controlled, but still we are seeing aberrant profile. So while applying this statistical outlier criteria will just help us to eliminate 1 out of the 6 replicates. But if, let's say, we observe 2 out of the 6 outliers, these statistical methods won't be helping us to, won't be suggestive to remove 2 of them, because even though it is predefined, but still we are seeing this, we cannot eliminate this outlier. So conventionally, as I mentioned, Dean and Dixon and Grubbs outlier tests won't help us in those cases. So it will lead to inappropriate estimation of the permeation results. So current approach for us is to predefine the statistical outliers in the study protocol, and based on the study protocol, once the study is completed, we estimate the outlier based on the statistical analysis by Dean and Dixon and Grubbs, define those outliers based on the statistical analysis, and perform the respective replicate and replace the data sets and perform this ultimate final bioequivalence evaluation, considering these outliers and replacing these outliers. However, from what we have understood, these outliers won't be a true representation of the permeation results. But at the same time, the current statistical approach also bars us from eliminating those. So this is another topic which we are pondering upon currently, if there is a way to navigate through this obstacle of outlier that would really help us to carve the path forward through this IVPT study. So yeah, these are the 3 major points which we wanted to share our thoughts upon. I'm thankful to my analytical leads who are currently leading the efforts, Priti and Srinivas, who are currently leading the efforts towards the development of new pathways for IVPT studies, and Amneal Pharmaceuticals, FDA, and the complex generics who have given an opportunity to discuss these subjects. Thank you.
I would like to ask our speakers to come to the panel, and I will introduce the rest of the panel members. So In addition to our speakers, we have Dr. Elena Rantu. She's a master scientist in Office of Biostatistics from CEDAR. She joined FDA in 2013, and since 2019, she's a lead mathematical statistician working with generic and biosimilar products. Her research work is mainly focused on assessing bioequivalence of topical and dermatological generic products, characterizing outliers in replicate PK studies, detection of data anomalies, and use of artificial intelligence and machine learning in drug development. She has a PhD from American University, Washington, D.C. And before joining FDA, she worked in academia and as a statistical consultant for over 15 years. We have also Dr. Rong Wang. She's currently the Supervisory Associate Director in Division of Bioequivalence 1 within Office of Bioequivalence at OGD. She has over 14 years of extensive experience within OGD, where she served as primary, secondary, and tertiary assessor of ANDAs, control correspondence protocols, and various generic drug regulatory submissions. And she has played an active role in various working groups. And she received her undergraduate degree in pharmacy from Shanghai Medical University and her PhD in microbial and biochemical pharmaceutical science from Institute of Medicinal Biotechnology from China. She also holds a PharmD degree from University of Florida. And last but not least, we have Dr. Narasimha Murthy. He's a Chief Scientific Officer at Topical Products Testing, LLC. He is a former professor of pharmaceutical and drug delivery at University of Mississippi for over 16 years. He is a pioneer in topical and transdermal drug delivery technologies, and he has published over 150 research papers and presented numerous scientific posters at national and international scientific meetings, and he has authored 2 books and over 20 book chapters. He has received several awards, which are listed in his bio, but the recent ones are the prestigious 2022 AAPS Pharmaceutical Global Health Award and also the 2023 award from Academy of Pharmaceutical Sciences in UK. He also is one of the co-founders of a nonprofit research organization in India called the Institute of Drug Delivery and Biomedical Research. So